Hello, everyone, and welcome to the Applied Research Spotlight for Statistic 3032 course. And I'm very grateful Myra invited me for this spotlight that I'm able today to discuss my own research and connect it to the ideas in the course. My name is Mariam Rahimi Mowassaq. I'm a PhD student in Industrial Engineering and Management Systems. Today, I will show how ideas and concepts from ANOVA can help us understand a real research problem in gene regulation. I will discuss Taguchi design and how it could help for future experiment and efficiency of the research. So here today the focus is just not the advanced biology or machine learning. I will keep the example of the gene regulation very simple, focused on the statistical reasoning rather than advanced biological and computational knowledge. So my main statistical story today has two parts. The first part is a significant result showed one approach improved the performance. And the second one is non-significant, which also helped me diagnose where the bottleneck was. And it does not mean when we do not get a significance, it does not mean that we failed. So I will also have that example to show different sides of the story. So here the goal basically is not to just fill in the tables, and we are using the statistics to make the research more defensible when we make the conclusions. So let me start with why gene relationships matter, so you be more familiar with my context. So here, the main research question that I had in my experience is, can a model recover the complete regulator team for a target? Let's see, what does regulation mean? What are DNA genes? So we will be able to understand the problem more concretely. DNA is could be thought of as the instruction book inside the cell, and the genes are individual instruction inside that book. However, our cells in the body do not use every gene all the time. A cell can turn some gene up, turn some genes down, make some of them quiet and inactive, and keep them that way until the genes are needed. So we also have some genes that help control the other genes. And these control relationships form a gene regulation network. So why is gene regulation network important? These regulation networks are important because they help us explain the cell identity, the disease behavior, and the response to treatment. So for instance, we have two cells. They have exact same DNA. You might be surprised that the DNA in our brain and the kidneys are essentially similar, but what makes them be different from each other is basically the genes and how the genes connect to each other. So we could have same DNA but behave differently because the different genes are active in these two different cells. So in this research area that we are starting to get more familiar with, the question is whether a model can recover which regulator genes work together to control a target. So It's the biological question is gene regulation, but a statistic question is how we compare the results across the experiments. So what is the key idea here? The key idea is regulation is a teamwork, and basically each of the regulators are not independently working on the regulation of the target. So we have groups of regulators work together, and it makes the tasks harder because it's basically identifying the individual regulators is not enough. So we have to recover the complete team of the regulators. What is the target here? Target is the outcome we want to explain, and we want to use the group of the regulator genes to be able to explain the behavior of the target gene. So the scientists, they observe the patterns in gene expression data, and they try to propose the team of the regulators. So now here we compare that the proposed team of regulators in this research, and we're gonna target that, compare that with like the real known group of the regulators working together. So let's talk about what is the goal of this spotlight. Now we know partially about this problem, that why it matters, what is the benefits of understanding the gene regulation. So now let's see what we are basically trying to achieve from this lecture. So this presentation that we have here is connecting the ANOVA concepts and partially the future extension of the Toguchi in this real research background. So we have two real results from my study that I would like to present. The first is a cooperativity results where our proposed approach improving the output over a standard approach. So we are comparing our model to a standard approach, and we are basically changing the factors that will be discussed in more detail. The second is basically the result is not significant, and non-significance of the results is helping me to understand where the bottleneck could exist in the model. So what is the core statistic question here? How do we use the replications to decide whether difference is meaningful? And that is the core idea of ANOVA. ANOVA is about understanding the differences and see if the source of the variations is basically, first of all, like understanding the source of variation and see if it's meaningful and significance. So we are basically comparing the structure differences rather than just looking, because we could have two different models. See, like this is like the output of this is bigger. However, to understand if it's significant, we need a structured way of the ANOVA analysis. So the second outcome that I basically tested in my gene regulation research is showing non-significance, and that also helps me to make the subsequent decisions about the model and where the bottleneck could be in the whole model. Then we will summarize and see like what we done in this presentation, what we discussed, and we also discuss a extension of Toguchi design, just as a possible extension for planning future study. And before we go to the statistics, let me define the gene regulation task in more detail. So, in the cells, our genes can influence each other, and target gene may be controlled by a small team of the regulator genes. So the research model is trying to recover that team from the data. So it produces a predicted regulator team, and then we score how close we are compared to the true label and the true team that are regulating, that we have it from the um, data itself. So you do not need to know the details of the model. We are keeping it simplified. You can think of it as a system that receives the gene expression data and returns predicted group of regulators. So target is the gene we are trying to explain through the group of controlling regulators, and the score that we get from the experiment is telling us how much we recover the true team. So we do have different metrics, but it's not necessary to know them in detail. But it's good to understand that we do not need to have just a single metric to measure the success of our model to achieve the task. So now we understand the problem, and now we have a very solid research question, which is, can our model recover the true team of the regulators? Now that we understand that, let's turn the research problem into statistical questions. So here, here we are basically we are trying to approach the statistical approach, like the statistical techniques that are needed to answer this question. And the main research question is established. Now we understand, like, we have to use our tools, statistical tools, to approach it. So basically, the statistical question here is the first step, that it becomes what factors is affecting the recovery of the correct regulator team. So in this setting that we have here, we basically will work on the factors, and the factors we will focus on are recovery approach. What it means, like, we have different strategies to recover the regulators. We have the different cooperativity level, which means how much we make it complex, how much we make the different genes interact with each other in the simulation. And we also have a different conditions of retrieval, which this will be discussed in the next slides. So the approaches we have are very straightforward. It's a standard approach versus our model. And cooperativity, which is a factor in our study, is basically how strongly genes act together. Retrieval condition, it means the model, is it using the normal retrieval or the complete retrieval. It's basically a special diagnosis setting. We force the true regulators to be in the candidate pool in our model. So we do have different response variables. We have recall, exact recovery, Jacquard similarity, which you do not need to know exactly what they are, but it's just giving a diversity in terms of metrics, and it gives us different levels of understanding how the model is giving the output. So in the first experiment, which we controlled the cooperativity, the size and the noise were fixed. So we are not using them as the factors here. So basically, in this particular slide, we had the main research question. We are quantifying it so that it could be measured and compared. So let's go to each experiment and see that basically what levels of factors have been used. We will define the factors and the levels, response variables, and the repeated, basically the repeat replications. Then we will have a statistical conclusion, and most importantly, the conclusion will be translated into research language and actions, because we basically run these statistical experiments to gain the knowledge to be able to make decisions. So let's go to the next slide, and let's define some statistical terms that they will be used, because we do not want to be confused here. So we do have the factor. What is the factor meaning in this presentation is the research condition. We are basically varying throughout the experiments. We do have different levels for the factors, and these are the options inside a factor. We have response, which is the measured outcome, such as the recovery percentage, it could be recall, and the Jacker similarity, which is an output specific to this research. It's not needed to be understood in depth, but it's just one of the metrics that has been used in the study. We do have replication, basically repeated matched runs with the same setting, we using random seeds and but shared conditions. So all of the conditions are similar to each, like in the repeated runs. The only thing that is different is the random seed and the computational conditions in that experiment. Statistical question here is basically in two experiments that I will be explaining. A statistical question is, are differences across level large compared to the repeated run variations? It means like between groups variation will be captured, and we will compare that to within group variations, and that gives us some answers whether the experiment has been meaningful or has not been meaningful. So now the experiment one design, let's discuss that in more depth. So the first completed experiment that I tried in my research was a cooperativity stress test. What it means is the statistical question was across the 30 seed cooperativity conditions. Does our model approaches basically the problem and produces the higher recovery than the standard approach? So we had the recovery approaches, two levels, our approach and the standard approach. And the factor two was the different complexity of model. How complex is the interaction of the genes with each other. It's controllable in simulated studies such as this one. So we had different levels, 0, 0.2, all the way up to 1. So we had 6 levels here, and it describes how much the data generation mechanism depends on regulators acting together. We had also response variables, recall, exact recovery, and Jacquard similarity. Each response is calculated for both approaches under the same conditions for basically making it possible to compare the results. So 5 random seeds has been used for each of the 6 cooperativity levels. So 6 levels multiplied by 5 gives 30 matched conditions. So each of the approaches produces one outcome. So because we have 2 approaches, it's 30 for each, and it gives us 60 model outcomes in general. So the comparison, however, is based on 30 paired differences. Because we are using 2 approaches to compare with each other, we have 30 paired differences, which we are testing to see if there is significance differences and if our model is performing better than the other. So basically the several settings were fixed in this experiment. For example, the size of the regulators was set to 3, and the count of our cells were like basically the samples were 2000 and the noise was 0.25. So these are controlled setting, and these are not factors in these particular examples. So let's see what we observed from this experiment now that we explained the factors, the outcome, and we understand the language that is basically taught in the course. So now we could have some insight here. So if you could look at the results here, it presents that the results were basically what I got from this experiment was significant. So our approach in this particular setting was significantly more capable than the standard approach and improved the recovery responses. Also, because we used 3 different metrics, and we basically were comparing, they are not independent from each other. So we use the home correction to make it possible to compare the results from each other, to compare them basically against each other. So for each of the metrics, let's look at the bars. For this output, which is also a response variable, we have Jacquard and 0.382 versus the 0.460. So there is a gain in our approach compared to the baseline, and the recall was also having some gain, which is the gain is 0.075, and for the other metrics, which is exact recovery, we had also gain, which the gain was in 0.060. So after the home correction for multiple outcomes, the improvement in 3 metrics were statistically supported. So the conclusion here is the modeling the regulator team as a set, as we approached in our basically in our method, it's improving the recovery under this controlled stress test. But we have to be very cautious about making the conclusions, because exact recovery, it increased, but it's still very low. So it's 0.113. It's not basically a high score. We improved it compared to the baseline, but we also have to draw very cautious conclusions. We should not say like our model solved everything. So another important limitation is the gain. Because we had different levels of complexity, our gain is not increasing monotonically with the level of complexity. So we should not claim that higher complexity caused a larger advantage. So what we could defend here in the conclusion is our model improved recovery across the evaluated matched settings. It's not that like its advantage followed a perfectly increasing trend. The gains are not basically monotonically increasing through the like different like increasing levels of the complexity. We have to be very specific when we are drawing conclusions, and we have to be cautious about our interpretations. So let's discuss what significant means, and we have basically, it's not simply a higher average. As I discussed, the importance of ANOVA is not just comparing, see like this average is higher than comparing to the other one. We basically had the R approach, which was set aware approach. It had higher average, but also it contained the variation across the replications. So, and we also had complexity, but we also had replications. So the question is here if whether the differences is consistent through the different replications. So we are basically not comparing. We have 30 conditions and our approach, 30 outputs on our approach, 30 outputs on the baseline approach. We are basically comparing two related group of 30 values, right? So we also have to understand that these variations, that we are comparing these two conditions, it is larger than the variation of the results through different replication. So it's very important to understand the ANOVA logic and how it works between groups versus the within groups to be able to draw the right conclusions. So both approaches here, they because both approaches, they separate the structure differences from background variations. So we often compare between group, within group variation. It's here like we had the method which was paired, and we had like a paired inference, but conceptually it's similar to what we are basically studying in the course. So now that we have the results, it was statistically supported, not just comparing the like higher level, higher percentage compared to lower, but statistically it was supported, and the conclusion is improvement was not just higher means, it was consistent across the conditions. So that supports a real method difference. Let's move to the second test. We didn't have the significance in the second test, but you will be surprised that non-significance also helped us to draw some conclusions and making decisions. So this is my second experimental design. Here we tested different sections of the model. So we had the first section of the model that suggests to us which regulators are candidates for the target gene. In the second stage, we narrowed this down to find the exact set. So now the question was, is the failure in the output caused by missing the candidates in the first stage? So this is very important to distinct that, because we want to see where the model is failing rather than basically just looking at the whole model as unified. So we broke the model down and we are asking, is the model failing because of missing in this candidates in the first stage? So here, basically, we had the different regulator sizes, levels of the regulator sizes 2, 3, and 4. And the factor is the retrieval condition. It means, is it perfectly the pool of the candidates, have we missed any of the potential regulators or not? Versus the no, like we have the perfect retrieval versus the normal retrieval here. So this also is a factor that has two levels, and the evaluation of the levels of the regulator size, and also the response variable exactly similar as the others, the first experiment. So same replications, we had 5 seeds, and each of the 3 are values, and if we multiply, it becomes 3 times 5, which basically, because replications are 5 and 3, basically are values, it gives us 15. We have also 2 different conditions, it's 30 outcome in total, but 15 pair differences, because we are comparing 2 different retrieval strategies here. So what is the perfect retrieval here is doing? It's forcing the complete true regulator set into the candidate pool. So we are making the first stage of the model perfect and comparing to the normal. See if it's responsible for the imperfect outcome we are having. So let's look at some of the values that we had. Like we could discuss the descriptive exact recovery, which is one of our response variables. And okay, so here we basically have the perfect condition versus a normal condition for the first stage of the model. And R equal to, it seems like the exact recovery increased very small very small portion, and at R equal 3 it's remaining the same. R4 equal to 4, the metric, the response variable, is also similar. So we were successful to fix the candidate pool, so we are basically making it perfect, so the model is not able to fail at that stage. So we see that the results are not different from each other. So it means, so we basically are descriptively looking at the patterns, and what we are suggesting here is what the problem is not this stage, because even if we make it perfect, we still have no changes in the outcome metrics. So that's what it is giving us. It's non-significance. It says we are not failing because of the perfect or imperfect retrieval. So it suggests the remaining problem occurs after this stage. So even non-significance gives us some decisions about our models, and we still have to explore the model and see where this model could fail. So now let's show the formal paired inference, and this is basically the non-significant results was significantly useful for our study. So the gains compared to perfect retrieval versus imperfect were not significantly different from each other. And as you could see, the gains are very negligible, and the 0 is included in the confidential interval. So that's suggesting that The retrieval, the perfect imperfect, is not explaining the failure in the recovery of the model. So here, as we discussed, is basically the main takeaway is a non-significant result can still be informative because it helps eliminate the potential explanations, as we did right here in this experiment. So basically, I would describe it as the evidence that this retrieval, perfect, imperfect, it was not the dominant bottleneck. And it's basically not evidence that this step does not matter. It's not that it doesn't matter, it's necessary, but it's not sufficient for improving the recovery, is what we could take from this experiment. So for the next slide, we will see what is the comparison of the retrieval loss versus the scoring loss here. So as you could see, basically this experiment is suggesting that the bottleneck is downstream from the retrieval, and our framework, which is trace, is the name. It tests that idea is tested more directly. So the question across the 15 matched CDR blocks, is a scoring loss significantly greater than the retrieval loss? So the factor is loss type with two levels, retrieval loss and the scoring loss. Response is the loss value calculated within each matched seed R, which was regulator size block. So the retrieval loss is basically defined as 1 minus the coverage, and we have the scoring loss, which is the coverage minus the exact recovery. So we define our losses, and then the experiment covering 3 different levels with 5 seeds and with 15 matched blocks. The decoding loss was 0, so it could not be responsible for any losses in the model. So the contrast is just a scoring loss minus retrieval loss. And if it's positive, it means that scoring contributes more failure than retrieval. So the formal tests is asking whether average contrast is above 0. And this was the whole of this experiment to understand if imperfection in one stage of the model is explaining the failure in the score, which was non-significant, which means this step was not responsible, and it helps us to get closer to where to look at the model. So here also we have the scoring loss and see the bottleneck. This is basically the domination of the scoring loss, which is confirming the bottleneck even further. We approached directly here that we had the 15 seed and different regulator sizes blocks, and we basically the scoring loss basically was exceeded retrieval loss by 0.504 on average, and so the entire interval is very well above the 0. It's indicates consistent positive contrast. So it's providing a strong statistical evidence that scoring loss is greater than retrieval loss. So it shows the same pattern, and this experiment, the contrast was positive for all different 3R values, and the study did not find significant evidence that the degree of scoring dominance changed across regulator set sizes. So the bottleneck pattern was consistent across the evaluated sizes. So the interpretation is very straightforward. The main challenge was not getting the correct genes into concentration. It was the step after retrieval, which was assigning the correct complete team a higher score than basically the higher score than the near miss sets, the potential sets. So the Oracle experiment, the previous one, versus the trace complement each other. These are basically the different sides of a coin. They are testing for the same thing through different, basically 3 different statistical experiments, and they both explain the same results. The same conclusion was drawn. It says like basically our failure of the exact recovery is not based on the retrieval loss. It was just based on the other steps that are after that stage 1 in the model. So now let's briefly discuss how Taguchi could be helping us in the experiment as a future extension, and then we will conclude and just summarize what we discussed. So here we did not test Taguchi, and we did not use it for generating the complete results that I presented. So the cooperativity test, the Oracle, and the trace experiments, they come from just a complete set of the combinations and the possible combinations that we had. So we are not showing Taguchi table here, but we still could discuss Taguchi as a future extension, because Taguchi is about efficiency of experiment. What it does, it basically gives us the contribution of different conditions, see how consistent they produce the outcome. Let's see, for example, our outcome will be recall in this example. So it shows us which set of this like replication in which experimental design are making like a high outcome, and that gives us some approach where to focus, that like where we have to like be more digging deeper in the model. So it's very efficient, and it gives us a tool to be more effective in terms of our costs of the computation, because sometimes in the research we have many different levels, factors within those levels within those factors. We have many different basically our models are very large and they're very slow, so we are not able to test everything. So Taguchi is giving us a lens because it's a balanced setting. It gives us for example, Taguchi L9 gives us 9 balanced settings rather than, for example, here we had 27. So repeating them also is multiplying by number of the seeds. It gives us more than 100. So Taguchi is basically giving us lower number of the runs. It makes it easier, as well as giving us a lens to see where we have to like dig deeper, because maybe it's not a magic tool. It doesn't replace the full factorial designs, but because it suggests reduction, it gives us like where should be focused, where should be like run the experiments more comprehensively. So this is one extension that could be there, is very significant, and we have to think about it in our research. I finally will close by connecting the statistical results to the main research question in the next slide. So we began our presentation with one main research question. Can a model recover a complete regulator team for a target gene? Then the later questions were statistical questions, and they were designed to answer specific parts of that broader problem. So first, the cooperativity experiment asked whether our approach improved recovery across replications compared to the baseline method. The answer was yes. All three metrics all improved significantly for multiple testing. Because we had multiple responses, we corrected by Holmes correction technique, and we still were struggling with the exact recovery metric, which was low. We did have gains, was significant gain, but it still was low. So the second experiment was asking whether guaranteeing the availability of the true regulator set would significantly improve recovery. The answer was no, because the estimated gains were small and the confidence intervals included 0. So that's non-significance also was useful because it ruled out a potential explanation for the low recovery level. We also had a backup experiment. It directly compared the scoring loss with the retrieval loss, and because it was very contrasting with this with the retrieval loss, so we had basically a confidence interval well above 0, and it confirmed that selecting and ranking the correct complete team was the larger bottleneck rather than retrieval. So what is the main statistic lesson here? Is that both significant and non-significant results can contribute to defensible scientific conclusion. Significance can support an improvement, while careful non-significance can also help us reject an explanation. Toguchi design remains a future planning option, not a claimed completed analysis in here. I would like to thank you for listening to this lecture, and I'm very grateful that I've been invited by Myra to talk about my research and its connection to the statistics course. I have one question for you to think about. In your own research topic of interest and possibly future career, what is the response variable, and what are the factors and levels within the factors? Thank you so much, and I hope you have a great rest of your summer and enjoy your vacation.